Swayam Prabha. Digital India. Educated India.
Need my recipe to I should have made it and made it on the well, I think that class. I don't want to talk about that. I think it was but I got sort of these okay, great. So I think we should get started and then we'll join later. So today will be the last lecture out of contents. Because tomorrow there will be other lecture will be like more focusing like forward so much about describing what system was first field, what will happen by pick up on some of the things that we can read or entertain in the future, both in the near future and the distant future. Which is the valuable start of that process, or like not only entertaining but also like will kind of promote your mind some reaction to some of the proposals about possible applications of the biology are really out of the box. Some of them are quite shocking, some of them may elicit some interesting feelings when you hear about them. Okay, well, yesterday we had this big lecture on circuits, and I'm sorry that some of you will not take it because I think that yesterday was one of the most important lectures of the course. I hope that you guys still see it. Stay, do you have access to the reports? Yes, they will really. And today we are going to tackle another big issue in biology, that is metabolic engineering. And I want to take the opportunity to use metabolic engineering also as a case in which you can see every single step that we have to solve, that we have to tackle when we face the challenge to develop a clinical state or even so in the field where everything covers tools, parts, models, circuits, metabolic and sensors, manufacturers, everything gets together for the sake of developing bacteria or microorganisms that do something. One little comment I want to make about this course is why we have been focusing only in microorganisms and in bacteria in particular. Well, number one, because we do not have time to refer to other systems that are already being studied under the umbrella for the perspective of synthetic biology, but also because similar to what happens when you study molecular biology, the very basic foundations of synthetic biology were established with bacteria. And therefore, everything about the part, the logic gates, all these things that we have been talking about these days, started to be applied in bacteria. Then the basic elements, the basic concepts, and the basic background for doing in another system were laid in bacteria. So that's what we want to do. That be aware that there's a lot of synthetic biology going on, mostly in plants, and then also in mammalian cells and in animal systems and so. But it is true that at the moment the field has not been developed so well as it is the case with bacteria. Okay, so just keep that. Okay, so let's start with some introduction to metabolic engineering. So just before we get to the concept, so just let me recap a little bit some basic concepts or some terminology in metabolism. I'm sure that some of these concepts and some of these terms are entirely familiar to you, as you have been studying this. How do we turn off this right here? Okay, good. So metabolism is a set of many reactions, but we can in fact divide the reactions into major classes. Reactions that have to have catabolism, the reaction with anabolism. So catabolic reaction or breakdown of a substrate. Okay? So that means that the bacteria get exposed to a substrate, and then out of it they generate energy, reaction power, and as a result, they produce small molecules and waste product. But the essence of catabolism is breakdown. Contrast to have anabolic reactions that are just evolutionary. They are all about variation. And because of these anabolic reactions, you have can extract large molecules and amino acids, acids, whatever. Three in this relationships, catabolic reactions and anaerobic reaction is that catabolic reactions will generate energy and reductive power, and in anabolic reactions you consume energy and reductive power. So this is something that then the input of metabolism is basically two is carbon and many some other minor components of the metabolism, but everything in metabolism contains carbon sources and nitrogen. So the carbon sources, the more habitual carbon sources are sugars, glucose, whatever, and some polymers. They're all like cellulose, some bacteria. The nitrogen may come in many flavors. One of them that is very common is the form of amino acids, and also the form of ammonia. And then in the process of metabolism, then if the input is oxidized, then you can start reduced product. And if the input is reduced, you generate an oxidized product. You can clear up all these possibilities. Some other work, there are basic blocks for metabolism that again you know already. So the main currency for energy is ATP. The main currency for reductive power or reductive or redox transactions is ADDH or ADDH that have this nomenclature. And as a minor player in this reaction, but also ADDH. So if it is not reduced form, you will find a element here or there. If they are oxidized, they use a there, you have a plus sign there. Okay, and then you have some reactions to which you can change one for the others. You can be phosphorylated, it can be sent to hydrogen and so on. So, but these are like the basic currencies that you have for each platform. For those reactions, the most important transformation is this one because this form of ADD, is the oxidized form, picks the electrons and then it becomes itself a donor of it. Okay? So, and they become these NADH, and the NADH goes in the direction to this electron. So, NADH or NADH are the major players of the electron recycling. If you are in an oxidized form, then you capture the electrons. If you are in the other form, then you excite them. Okay, so the other general picture that you may have in mind for metabolic engineering is this idea of cell as a catalyst. So in general, you have a cell, and the cell can be for the most part bacteria, but it can be other as well. And you have inputs here that the cell deals with as a catalyst. You have outputs. By default, the outputs that you get are two molecules. You have some proteins, you have large molecules. Okay, so also before I get what we call temporary engineering, let me just share with you some empirical perspective that perhaps you know already. But technology has been around for a long while. Also, it's a secret for production of open greens for fermented food. And well, even without any knowledge of genetic engineering or DNA or anything, just by trial and error and pure technology, you can really do a lot of things. And in many cases, even before the era of recombinant DNA, it was possible to produce some compounds at large scale just on the basis of the biological matter that were behind the production. But then, the big change in double engineering is that you enable that technology to be based on engineered biological systems, not just in existing biological system, and that it enables that you understand the process, what is behind the process, what is the mechanism of the process, instead of being just an observation. So today, the chemical industry is a big industry, is the basis of everything we see around, all the materials, all the liquids, many of the things, and it's a super big sector in our society and in our economy. The problem is that the most part chemical industry deals with non-renewable resources. If the resources are provided by areas of the world that are predictable, and also the chemical industry typically produces the waste. So these are the downsides of the chemical industry that, on the other hand, is the basis of our society and economy. And much of the chemical industry has to go with some petroleum expression, and therefore, for many years now, it is to see what degree instead of using chemical procedures that are contaminant, developed with petroleum, or have to use petroleum as a feedstock, or is highly contaminant, whether we can replace these chemical factories and this massive catalysis that happens in chemical factories by something similar, in which main agents, the main catalyst is an engineering material. So the idea would be to start with some materials, give it to the catalyst, and then introduce some products with value, chemical products or other products. The advantage of using bacteria or microorganisms as catalysts is that the material itself is so you don't need to do it every time because it will by itself grow and replicate. From an environmental point of view, in principle, is more environmentally benign, so that means that it's less impactful in the environment, and also in many cases, the starting material are renewable, cellulose, residues of different reasons. So, in principle, it's a very, very attractive situation. Then, instead of having these big factories, maybe we will have to concentrate in considering bacteria or cells in general as factories themselves in miniature, where you have some equivalence in the structure of the bacteria and what you see in factory. You have a relocation mechanism, for example, you have an assembly line, a kind of central computer that you have inside the cytoplasm. You have outer and internal walls to protect the factory, and even you have a security fence that covers the whole. You can make some kind of equivalence between what you have now. Bacteria, you have microbes. So please consider bacteria and microbes as a small miniaturized. So what can we do? What products are produced upfront with metabolism? Well, these are the most, what we call the low-hanging fruits. They are that we discuss some of them tomorrow. But if the idea is to replace chemical processes by biological counterparts, the first type of products that we will consider are those that are typically produced by chemical industries, for example, foods, whatever, engineered products, plastic, polymers, some special chemicals, and some cases also some complex molecules, vitamins, pigments, and so on. So in many cases, bacterial systems or biological systems solve a problem that chemical chemists have very many difficulties to solve. and it's the chirality of some molecule. So what happened that because of this chirality and symmetry problem or issue in molecules, then it may happen that the molecule are active at the origin, whereas the bacteria persistence are really specific in production one of. So this possibility of solving the chiral component in molecules one of the advantages of using bacterial biological catalysts. Okay, so the history or the process that leads us to contemporary metabolic engineering, we can distinguish also some early attempts of using genetic engineering for this type of applications. And in the early stages, it became very clear that with some just a few, say, tricks coming from the recombinant DNA technology, we could really improve the production and the possibilities of using bacteria for different applications. So what we had to do at that time was to choose a host cell to identify DNA or some genes that encoded a given phenotype, inserted into a DNA vector, put in the vector in the host cell, isolate the cells that receive the vectors, and then hope that it will work, and then to become, by that time, metabolic engineering as well. As you will learn in a few minutes, is not the case. But the early development of metabolic engineering dealt mostly with the application of the actions that you just heard on a limited number of hosts. By that time, they were not called a chassis. The chassis came much later under the umbrella of biology. So we had E. coli, we have Saccharomyces, bacteria, other bacteria, then again, so depending on the final application that was envisioned for that particular catalyst, then the host of the recombinant genes will be different. E. coli was for many applications, Saccharomyces for ethanol, bacteria for enzyme, and so on. So I have to praise for a few slides E. coli as a very well suited organism for metabolic engineering, despite the fact that it has some shortcomings. So E. coli as a model organism for these applications has been super successful and is still to this day is the platform in which most metabolic engineering endeavors are fully practiced. So why? Because it grows very fast and can be manipulated in very, very well. The metabolism is very well understood. It produces natural organic substrates. It grows in various carbon sources. The media where it grows is very simple. The metabolism can be manipulated easily. And also, if you make some models, the theoretical genes that you can get after making some manipulations in metabolic engineering are very so even though my own laboratory is very keen to use Pseudomonas stupida and later to use some applications of metabolic engineering with Pseudomonas stupida, in reality, E. coli remains like the king of metabolic engineering that is surrounded by other bacteria, yeasts, and other platforms for this type of things. But to this day, no one has been able to beat E. coli as a preferred platform for metabolic engineering. Reason for that, and this is one of the reasons, is because E. coli is endowed with a high metabolic plasticity. It can take a large number of substrates, in particular sugars, and some acids, and the central metabolism, it is able to convert these substrates in a variety of compounds that in some cases act as building blocks or in some biomass, like other cases are just secreted to the medium, and that you may identify here. Some of them look like interesting molecules for the clinical use. Note that some of some molecules are pure. Plus to minus, then that gives an advantage with respect to purely clinical procedure that we need a racemic mixture of whatever to. Okay, so let's see what has been the major transition between what we call or we call early metabolic engineering and then later metabolic. So in tools Then the idea was to random mutagenesis, heteroexpression, pathogization. Then later it was possible to increase the of this method by more targeted pressures later. And then finally, all change had been the possibility to execute what we call systems level engineering. So that means that instead of a concentrated one or two or two genes, then the idea now is to consider the entire metabolism as the subject of study. And this is possible because the last decade or more, that has been possible to reconstruct entirely the whole metabolic network of E. coli as well as many other bacteria in such a way that we have a very precise idea of what is the fate of a given substrate when it goes into the metabolic network of E. coli, and then we can predict what will be the fate and the final destination of every carbon that is in the substrate in every product that has from passing that substrate to the whole metabolic network. Okay? So, and then E. coli is the bacterium where you have a very favorable connection between the metabolic network and then each of the genes that are involved in the metabolic network. So, as I said, the E. coli to this day is associated together with Saccharomyces cerevisiae, but I will not deal with that. And to this day is the best selling platform for this application. So, and this is possible because there are various models increasing in detail of for the metabolic of coli. Well, one of the best was produced in 2007. And what it makes is to make all the connections between all different metabolic reactions and then give them different values, different parameters. And then if you decide to enter into the system as a here, the system will tell you where it goes and what will be the distribution of the different cargos in various locations. And if you have some experimental indications, you can even predict what will be the fluxes of the different cargos of the different substrates going to the whole same edge. So this is something that for metabolic engineering, metabolic engineering is very important because it allows you to plan things much before in a computer, before you really start doing the actual experiments. And then the system will tell you a kind of a score of what would be the success, the perspective success that you may have with a given metabolic engineering operation. So some of these models have many reactions. They are annotated in correlation to different works of different genes. A large number of metabolites have been identified. There are thermodynamic information for chemical reactions and so on. So today, any metabolic engineering process starts by sitting in front of a computer and seeing exactly what one metabolite wants to produce, whether that metabolite or what that molecule can be produced naturally or can be extracted from the existing network of reactions that we have E. coli for original bacteria, or whether we need to add some genes from other origins to make that happen. Okay, so this is possible because there are more and more scale models of every bacterium, and then one can pass for trial and error, tinkering, trying here and there, and see what happens, to have a completely rational approach or very, very highly rational approach to this metabolic engineering. One big tool to this end is the availability of, say, platforms that will tell you or will predict what is the consequence of knocking out a gene here and there in the final distribution of metabolites in your strain, or in particular in E. coli. So years ago, for instance, these three platforms were developed of gene and of strain. And in particular, the first one has been quite useful because it will predict what will be the consequence of making specific divisions in the performance of the metabolic network. And this is something that is very nice because you upfront, you know exactly what's going to happen when you make given decisions. And then by doing that in silico, you have a good indication of how the system may be later. And what you have other platforms here, and in particular, the strain will tell you what will happen if you enter the strain pathway that comes from different organs. How can you couple the impulse that can be channeled through the new pathway into the central metabolism? So these are some of the examples. There are many these days of platforms that help the metabolic engineer a picture of how even project new results even before you start making the experiments. For instance, this is just one example, a couple of examples I will show you. With this one of these platforms, one can predict what will be the consequences of some knockouts or production of a specific metabolite. So in this case, well, this is very well, this is a project for generation of saxin. And then the system tells you that actually here, there, then you have more or less amounts of saxin being produced at the end. So at the end, because of this indication, you make the meters, and then you have a good chance that the engineer strain, by introducing that mutant, will produce more saxin than in classification. So you can either examine the effect of including targets, the map and say, I make a knockout here, chances are that the fish in this direction will move to the other, or you can model in a rigorous way what will be the effect of the mutation without having to depend on your intuition. Similarly, there are many papers, many applications in the literature showing how specific mutants here and there may result in overproduction of given metabolites. For instance, in this case, it was an operation a genetic intervention in E. coli to make it be a hyper producer of L-valine. So every amino acid can be produced by separate, and they all have a high value for various purposes. But in this case, if the authors wanted to make a strain for hyperproduction of L-valine, then the system will tell them what parts of the pathway that goes from glucose to valine they may want to knock out to increase the flux between the substrate and the product, and avoiding all these kind of side reactions that might interfere with the final production of the stuff that you are to. Therefore, we are in a situation in which it is now possible to go from the beginning of having random mutations, but now we can make targeted mutations and then combine these targeted mutations with predictions that we have a computer, and LCN can really deliver things that do much better than they do 20 years ago. So this will be the final picture of the pre-modern metabolic engineering, in the sense that we have targets, then we can combine various bits of information, we are compiling, and at the end you produce an engineered strain that is the final result of the metabolic engineering process, and then you pass the strain to your engineer friends that will be able to upscale and to convert that property that you're seeing at the level of a small reactor or at the level of the finish into a big reactor. Okay, so for construction of more complex pathways, not just knocking out here and there to produce amino acids or metabolites, but for construction of a complex, say, molecules where you need to bring in genes from various origins, then the process can be helped with, again, a number of platforms. For instance, you have this very famous platform that is called the Biocatalysis and Bioregulation Database. This is called the Minnesota Biocatalysis and Bioregulation Database that is only in Minnesota any longer, but the name has been taken. That is really interesting, and to some extent is present, because the inputs that you have to give is what you want at the beginning and what you want at the end. So I say, well, I have glucose, I want to convert glucose to insulin. And then the system will survey all databases and will find various pathways that should be able, under ideal conditions, to convert that substrate that. And on top of that, it will also associate every state of pathway to literature that will refer to specific things that can deliver that reaction, regardless of which the original process. So this is a super useful resource. It has been used by thousands of different scientists to listen. More recently, there has been this other pathway here. Well, I did that. I see you start to see what that's another. Okay, so this is another big platform, more recent, by the Pacific Mechanism. That this is something very, very well set up. Basically, it's a platform that will put in front of you every single biochemical reactions that have ever been seen and that have been ever, say, observed, and that can be also be combined in a different way as they appear naturally for the sake of metabolic engineering. So it's like a super repository of reactions. I say, well, I want to convert substrate A into product B. What should I do? So the system will go through all the literature, all the databases with biochemical data, and will tell you what reactions you have to implement for converting a given product and a given substrate in a given product. And in this case, the advantage in connection with respect to the previous platform is that it will also calculate not only the reactions as a kind of qualitative problem, but also the thermodynamics of each reaction. So it's such a way that it will tell you not only if the reaction is will be happen, but also whether it is thermodynamically possible, because this is something that other platforms do not show. So it's an amazing tool, and our engineers are very happy to having this tool available, even though you have to register and has a beta version and a version, but conceptually it's a super interesting.
So, yes. So in case in Atlas model, the dotted lines represent what is, I mean, there is theoretical reaction, it could happen, or it's getting from some.
Okay, so well, in case basically what you have is a list of reactions and a list of products, and how in the instant metabolism you can go from one product to the other. But then the advantage of Atlas is that it doesn't tell you only the reaction that exists, but also the reaction that could exist. So it's another kind of structure. And not only that, but it makes us a thermodynamic calculation of whether, I mean, you may have an enzyme that can act on a substrate, and then on paper it has that activity, but will it work in the context of metabolic network with all the thermodynamical constraints that we get in constraint? But this is something that Atlas will tell you. And this is one of the advantages of this platform as compared to the other. Okay, so where we are at the moment. So metabolic engineering is evolving more and more into a system development approach. More organisms, not only E. coli, are becoming accessible. So some organisms with metabolic traits are becoming also interesting with metabolic engineering. More and more genome-scale metabolic models. We know also more and more how to handle regulatory networks, and we have a good example in a moment. And also, I have to say, as I mentioned before, that metabolic engineering is becoming a big thing from the point of view of the public and economic and industrial interest, also because of the communities and everyone says that that at some point we will run out of petroleum, and when we run out of petroleum, we have to look for other alternatives, making complex molecules, and for the time being, metabolic engineering is the one technology that works. Okay, so well, what I told you so far, I have a grand view or grand background of what metabolic engineering may look like. And well, let's go in some more detail and show you some examples of how far we can make with metabolic engineering, and how how many technologies we have to use to implement metabolic engineering projects at this time. So, metabolic engineering basically is about to win the existing regulation, the existing metabolism of bacteria, and then convince the bacteria or to program the bacteria to do something that not necessarily is in their interest, is more to our interest. So, and therefore, we have kind of fights with the biological system in the sense that the biological systems have their own agenda that in many cases is not coincident with our agenda. However, because of the tools we have in many cases, not in all cases, but in many cases we can program, we can kind of convince the biological systems to behave in the way that we do. So and then it can be applied to cells, but also to cellular organisms or products. And as I mentioned before, producing biological approaches has advantage in respect, in particular, in selectivity of that's produced because of the biological characteristics analysis and well, the factories and academic activities and produce chemicals. You have to go back for different possibilities about by all the tools that we have discussed in previous days when applied to specific projects or metabolic engineering or so, as I mentioned before, bacteria offer some advantages in respect to the cells. They are small, they reproduce very quickly, they have a great diversity, and in many cases, we can isolate bacteria that are present or can survive in extreme environments, and also they use nutrients that are not available in other organisms. So one of the kind of grand views of metabolic engineering is to use waste and convert waste into value. And this is something that so far only bacteria can do in ambient environment. So there are some downsides. The main downside, as I said before, is that they are evolved to do their own agenda, and therefore they are evolved for surviving, not for doing what we want to do. And in many cases, they are optimized for low nutrient levels because in the environment they don't have plenty of nutrients. They have different systems, and they naturally don't make, for the most part, the things that we need. And therefore, in many cases, the idea is to start with an existing bacterium and then redesign the existing bacterium to optimize it for even for meeting specific industrial parameters. And it involves in the engineering of the bacterium to not only to redirect its metabolism to produce what we want, but also to stop them from making things that we don't want them to do, like retaining products, like secreting products, and like producing the systems that interfere with the biochemical process. So it is possible, and this is something that historically has been done in some cases, to make some what is called irrational design. So irrational design is something that is simply that you start with a bacterium, or this is a fungus, that does something interesting, like production of penicillin, and just by subjecting the microorganisms to chemical agents and then selecting for those that will do better, you can really induce a large number of mutations in the microorganisms, and you have a way of selecting the ones that you can really increase the productivity from a few milligrams. That was the case in the original penicillin strain to really produce a lot of penicillin up to 1,000 more increase just by doing this mutation where you really don't know how it goes. However, this is time consuming, expensive, and at the end you don't know exactly what is the reason for this production. So there are since early in time there were approaches to go from this completely random approach to go to something that is more rational and more efficient, such as, for instance, genome shuffling. So genome shuffling is this idea that you take bacteria from various origins and you interchange, you kind of shuffle the sequences of the two bacteria to generate a tremendous diversity. And then if you have a way to select for those variants that do the job better, then you have a good tool to increase the production or the productivity of a given process
without having to go into this generic mutagenesis that you don't know. So in this slide, it is compare how what you may call blind mutagenesis can give you some little by little, say, increases, and in many cases, in some cases, the old way it has worked. But with DNA shuffling, then in some cases you can have like big jumps in the productivity of a given pathway. Okay, so that's something that was very good. But the ideal thing would not be to have a kind of shuffling or general approach, but to be more specific and target specific modifications for the production of something that you are interested in. Okay, so we can do that because we have not only tools to do it from a weight point of view, but also we have a lot of weight tools and weight, say, methods to introduce in and out genes, foreign genes, in a cell, in a platform. In many cases, E. coli, but in other cases, other bacteria. Okay, so every single genetic tool that we have been discussing in previous days can be taken now for the sake of metabolic engineering in E. coli and similar bacteria. Okay, so then the starting situation is always something like this, where you have a super complex metabolic network, but then thanks to computation and to the models, we are not lost in this mess. We can really focus in one particular part of the metabolism and then figure out what we have to do if we want to hyperproduce this metabolite, because this metabolite might be interesting from an industrial point of view, or we can figure out how we can take bits and parts of the metabolite or the metabolism to increase, to be a better host for a new pathway. This is a starting point where you have to intervene for making the product of your choice. Okay, so I'm going to spend now some time telling you, because I think this is the archetypical example of a very deep metabolic engineering case, a metabolic engineering example where you can see everything that is necessary to really have a bacterium that makes a tremendous difference from the point of view of the biotechnology associated to the product at stake. And it's the famous or infamous artemisinin case. So let me make some background. And the background is that there are many molecules that are called terpenoids that are super interesting molecules from many points of view. Some of them, many of these products are made by plants. So that means that the pathways for their production are not in bacteria, but some of them have very important economic value. For instance, menthol, or some chemotherapeutics, or lycopene, or Taxol. You see that all these molecules share some chemical motifs, and in many cases, the idea would be to produce them not in their natural context, because they can be difficult to obtain, but trying to express them in other parts. So the pathways for production of these terpenoids in many cases are known, but they are known in plants. They are not known in bacteria because the bacteria don't produce them, right? So you go to the plant to the databases of plant metabolism, you find many enzymes that this has been a bit offset, but in any case, this shows that you can go from some molecules to the others, and you know the genes that are involved in the plant, and at the end you produce terpenes, sesquiterpenes, and the terpenes on the basis of precursors that are common to different types of biological systems. Okay, but the genes of interest are always in plants. Okay, there is one pathway in particular that is quite interesting that starts with pyruvate and ends in this compound here. In these two compounds here, they call IPP and DMPP. That, as you will see later, are important precursors for this final molecule that we want to obtain, that is artemisinin. Okay, and also in E. coli, there are bits and parts of this pathway, that is the mevalonate pathway, that at the end produces also some of the compounds, some of the intermediates that will be necessary for production of this target molecule, that is artemisinic acid. So the situation is that in E. coli, we have some bits and parts of a pathway, but other bits and parts of the pathway have to be found somewhere else. Some of them in yeasts and some of them in plants. So the idea is how we put everything together for the sake of production of this compound that is artemisinin. So artemisinin is a very valuable molecule because it's an anti-malarial compound. it's used today as a medicine, and is one of the best molecules that help to control malaria. So the original origin of not the original origin, but the origin of this compound is this a plant that's called Artemisia annua, and for centuries it has been known that this plant, or its infusions or its extracts, had something that was really good for combating malaria infections. Okay, so there's something important, is that even though the final medicament, the final drug is artemisinin, in reality the artemisinin comes from a precursor that is called artemisinic acid. Okay, so once you have artemisinic acid, converted artemisinic acid into artemisinin is piece of cake. Chemically can be done with no problem. But the difficult thing is to get this precursor in a system that is not the plant, but is a heterologous expression system. Okay, so the bottom line is that the cost to produce a dose for a patient is, or by the time that the slide was made, was to $40. But this is very expensive, and calculations indicate that to make the system available to a large population, the price has to be like 10 times less. So, there's some more data on the economics of this thing. So, if you keep the price of the drug at $225 more or less, then many people will not be able to have it, because in some countries the expenditure of in health for a person per year is very, very low. Okay, but also it's true that an infected person needs about 12 treatments per person a year, and then well, simply it will not work because of the economics, right? And the WHO suggests that we need, or the planet needs, at least to produce 700 tons of the artemisinin to cover all the necessities for patients for malaria. Okay, so there are two ways of producing artemisinic acid. One of them is cultivation, and obviously you have to have big extensions, agricultural extensions of land that are planted with the plant and everything, and then you extract the molecule, and then at the end you produce artemisinin, and that's the end of it. But the idea, the ideal thing would be to produce it through fermentation, with either a yeast or a bacterium, but that at the end, instead of having to go through all this agricultural, say, mess or agricultural pipeline, you go to something that is more concentrated and more productive in many respects. Okay, so then here comes the idea, or the problem that started to be entertained in the early 2000s on how to produce artemisinin in a simple E. coli-based platform, because E. coli is, as I said, one of the best platforms for this type of things. So obviously, microbial fermentation will be ideal, because you can have them not only in agricultural land, you can have it in many places. And also the starting materials could be inexpensive. You don't need a land for seeding or much work. But then you need a whole bunch of, you need to overcome a whole bunch of challenges. Obviously, you have to make sure that all genes of all the enzymes in the pathway work. Sometimes expression of eukaryotic genes in bacteria don't work very well. You need also to balance the expression of the different bits and parts of the metabolic pathway for optimizing production. And also you need a good platform organism, in this case, E. coli, with all types of tools for doing that. Okay, so this represents in a graphic form everything that is required for the engineering of this pathway in E. coli, okay? First, you have to identify and pinpoint and sequence, well, no sequence, genes are sequenced in many cases, but just to extract the sequence from the plant, okay? That will be one of the challenges. Then you have to clone the genes and put them back, or put them in E. coli, and secure that they are expressed at the levels that you want them to be expressed. And for doing that, you need to control gene expression with many of the tools that we have discussed in previous days, with promoters, with circuits, with terminators, with Chandel-Garnos. because the signals that bring about expression of the genes in the plant are completely different of the signals that bring about expression in E. coli. Okay. Then you have to control the supply of intracellular precursors and wait. Yes. Okay. What happens here? Okay. And then off you go. You have to go little by little. Well, in this project for the artemisinin, you will see in the next few slides many things that you have seen in the past in general, but now applied specifically to the problem of developing an optimal pathway for production of this artemisinic acid that eventually will be transformed in artemisinin. That is the final drug there. Well, first of all, you need plasmids to make the chlorine and everything. We have seen many of them, and that's in principle is not a problem. However, there is a little detail here that has to be considered, and is that not all plasmids are equally useful for metabolic engineering. Okay, because if we have an existing pathway here in the strain, a default pathway with some of the precursors and so on, and then we enter one new enzyme in a plasmid, whether the plasmid is high copy or low copy may make a big difference. Okay, so sometimes having one enzyme clone in a multi-copy plasmid and entering that plasmid in E. coli makes that enzyme is produced at very high levels, and that might not be good because then you unbalance the whole flow of metabolites into the final product. Okay, so one of the variations that you have to consider or had to be considered to optimize this situation is to deal with either a high copy number plasmid or a low copy number plasmid. In this case, as is represented in the cartoon, when you have that enzyme in a lower copy number, it seems that the cells are happier and you don't generate such a big disturbance or disruption of the channel that you have at the end. Then you need promoters. Okay, you need plasmids, you need promoters. In previous days, you have analyzed many of them. And just for some details, remember that not all promoters work the same, and not all of them are equally good for this type of applications. One super typical promoter that is used in E. coli for metabolic engineering endeavors is the arabinose inducible promoter, the so-called PBAT promoter. Okay, and this is a very popular system for heterologous expression of genes in E. coli because it's very it responds very well to the inducer, to an inducer that is arabinose, and not only that, but it also happens that the response is very linear. Okay, so the idea is that you have the bacterium with the genes that you want to express, and then all you have to do is to add arabinose, and then at the end things work. This promoter has been characterized. These promoters have to be characterized before they are applied to expression of heterologous metabolic genes, because there are some characteristics of the promoter that you need to know upfront for having a perfect and optimized system. But in any case, in the case of this promoter, then you have a very good linear response in terms of fluorescence with the GFP and also the arabinose concentration. So this is the ideal situation for a metabolic engineer, that you have a linear response of the output of your promoter in respect or in response to growing concentrations of the inducer that you have in the system. Okay, so that's more or less what you want and what you have. But I have to make a little parenthesis here. And the parenthesis is that in this course, we have talked a lot about controlling gene expression with transcriptional factors and with repressors. Repressors, activators, inducers, and all the rest of it. There is one, a method to control gene expression that is not based on transcriptional factors. It's based with another biological, phenomenon, biological material that are called riboswitches. And let me make a little parenthesis and tell you and show you a few slides about riboswitches, because the entire panoply, the entire toolbox of a metabolic engineer or a genetic engineer includes riboswitches, a part of having also expression, say, systems based on transcriptional factors. So I will spend just a few slides to tell you what riboswitches are. So the basis of riboswitches are the so-called RNA aptamers. So RNA aptamers are RNA molecules that acquire conformations that are able to interact with a small molecule, okay? So you have this RNA, for instance, and then the RNA may fall with a secondary structure, but then there are parts of the secondary structure that may not stick to each other or may not pair with each other, and then this creates a sphere, a kind of interaction sphere that can accommodate a small molecule, and that small molecule may either stabilize the structure of this conformation, or it can destabilize it. Okay, so that is called an aptamer. So an aptamer is a small RNA sequence that binds or doesn't bind a given small molecule, and depending on whether binding occurs or does not occur, then the RNA structure may acquire a conformation on another conformation. So this is something that happens in some cases in a natural form. For instance, there is a very well-known aptamer that binds theophylline. That's a well-known, compound. And also, it is possible to generate these aptamers in an artificial way. You can generate a combinatorial library of these bases here, and then you can check whether one of the combinations will bind a molecule of your choice. Okay, so that's something that is very good. And the idea here is that most aptamers will exhibit conformational changes upon binding to a small molecule, because it will be a fit binder. Okay, so there are natural aptamers, as I said. Many are prokaryotic. In other cases, they are eukaryotic, even though in the prokaryotic world is when they have been better characterized. And if you think about it, it is possible that if by this interaction between the RNAs and a small molecule, the DNA conformation changes, it is possible to combine this change with the expression or not, or the availability for translation or not of a given mRNA. Okay, so that's the basis of what is called the riboswitches. So riboswitches are the result of putting together an RNA aptamer with a longer RNA in a fashion such that, depending on the conformation of the aptamer in vivo, then the RNA might be available or not for translation. So basically, the idea is that, and this is an interesting level of regulation, because it's completely independent of transcription. Okay, so you have transcription, okay, but then, you know, if at the start of the RNA you have here mRNA, and then you have your genes, gene A, gene B, gene C, whatever. But then, if at the beginning of the mRNA, you have a RNA structure that interacts with a small molecule, and the ribosome has to land there for starting translation, then the small molecule may allow or not allow availability of the 5' end of the RNA for translation. So that means that we can generate a situation in which a small molecule can control translation of the RNA, and therefore what happens at the end, you have a control of gene expression in a fashion independent of transcription. Okay, so that's a nice thing. And also the main advantage of the riboswitches is that as they are based on RNA, synthesis of RNA is much easier than synthesis of any protein. So you can have a kind of orthogonal control system of gene expression without having to be concerned about proteins. Well, in any case, so there are, as I said, some aptamers out there that are natural, but they can be produced also in the laboratory. Okay, so for instance, you have some of them that are very well known. One that is able to interact with a molecule, and then this small molecule here will generate a constellation of interactions between different reactive or different functional residues here and functional residues in the RNA. So whereas in the case of transcription, you need to have a repressor or a transcriptional activator to bind or not bind DNA, and then this binding can be interacted or can be influenced by different effectors. In the case of a transcriptional, in the case of a translational riboswitch, what you have here is just, as I said, the availability of the RNA for translation, depending on whether or not a small molecule is interacting with this DNA structure. Okay, so you know that this gives us another level, another possibility of controlling expression. As a matter of fact, in some laboratories, there's a famous laboratory in Stanford that argues that in synthetic biology, the only type of regulation that we should use, because it's absolutely orthogonal, is the one based on RNA aptamers, because you don't have to deal with transcriptional regulators or with any other things, only this type of riboswitches. But in any case, one can combine, as a kind of practitioner, both the regulation engineered on the basis of transcriptional regulators together with the regulation based on riboswitches. Okay, so this could be the kind of design. Okay, what you do is that instead of designing a binding site for or to put a promoter element with a transcriptional factor or whatever, what you do is that you take your mRNA, the mRNA of the gene that you want to express, and then in front of the RNA, you engineer a sequence that is based on RNA and that will bind or not your small molecule, and therefore by doing that will enable or not enable translation of the sequence here, and therefore will enable or not expression of the corresponding protein. Okay, so that is also very nice. So this is very good when you have to express in a bacterium genes that come from a completely different origin. And because in that case, the only thing that you have to do is to, as I said, to put in front of the structural gene that you want to express this little sequence there that will by itself produce a transcriptional, will produce a control of the gene expression here in a fashion that will still depend on an inducer that you add to the medium. Okay. Well, there are many riboswitches out there. As I said, some of them are natural, others can be engineered. And even though the regulation that one obtains has sometimes a narrow, window of effectiveness, reality is that it is possible to use it, and many laboratories use that for various purposes. So some riboswitches are well known and so on, and others, as I said, can be engineered in the laboratory. There are various mechanisms involved in the control of gene expression through riboswitches. I mentioned some of them here. They are all based in changes in the conformation of their RNA structure when the RNA structure binds a given metabolite or a given inducer. So the main mechanism is translational control, but it's also true that because of the formation of secondary RNA structures, then these riboswitches can also be engineered to affect termination and also to affect RNA processing. Well, I don't want to get you into more details, but just please remember that a part of transcriptional regulation based on factors, on protein factors, you have another possibility to control gene expression based exclusively on RNA sequences. And that's something that is very good. Well, I will not tell you there are a bunch of possibilities here. And well, just for the sake of this description of possible tools that one has to optimize a pathway, just remember that riboswitches is a possibility. Okay, so then we continue with the story that we were discussing on how to optimize the pathway for production of artemisinin. Both in the case of transcriptional factors and also in the case of riboswitches, you have this phenomenon that you have to be aware of and that we have also discussed in previous talks. And it's about the fact that when you induce gene expression, then it may follow two different possibilities. One of them is that the entire population is induced simultaneously, and therefore induction or expression levels are simultaneously coordinated in all the population. Or remember that we have also the possibility that we have a lot of noise. A lot of noise means that not all proteins, not all cells are induced at the same time because of the promoter. But the thing is that you move from no induction to full induction in a population-dependent fashion. Okay, so remember that we discussed this with some of the cases that we have examined. And if you detect expression of a specific genes, if the gene, for instance, give you a fluorescent input, a fluorescent output in a fluorometer, then you can monitor how expression of individual cells happen in a population. And then remember that we have these situations where we may have a bimodal, regime that then cells change from a non-induced status to a fully induced status without having anything in between. And it happens that this is something that happens often, that instead of having this sequential increase in the expression of the gene of interest, what you have is this other kind of jump between one or the other. And that in that situation, in many cases, also influences the type of response that you have in your system. This is another consideration that one has to take in mind, that we have to keep in mind for engineering pathways, whether you want a promoter that will deliver a continuous induction, or you prefer a promoter or an expression system that will give you this bimodal behavior. Another consideration, another choice that you have to consider. Okay. In the case of the Arabinose system that I just mentioned, the thing is that it's a bimodal, induction. So that means that the cells go from no induction to full induction with nothing in between. Okay, that might be useful for some cases and may not be that useful in other cases. Okay, but then we return to how we apply all this panoply of different possibilities to production of artemisinin. So artemisinin is about the pathway for production of artemisinin, is about cloning the genes, expressing the genes, and then optimizing gene expression. But this is something that we'll see after a 5 minutes break, okay? So please be back in 5 minutes.